Good morning and good afternoon. Thank you for joining day 2 of our 2020 virtual user meeting, Membrane Physiology Beyond Ion Channels. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentation. By default, all audience members are muted until the end of the presentation, at which time we would open the floor to some questions. If you have questions throughout the course of the presentation, we welcome you to comment in the chat box. It is now my privilege to introduce Dr. Matthias Quick of Columbia University in New York. His presentation is entitled Employing SSM Electrophysiology to Capture Electrogenic Fluxes in Membrane Proteins. Upon completion of the presentation, we'll proceed into the Q&A. And once again, we welcome you to submit questions in the chat box throughout the course of the presentation. I will now hand over the talk to Matthias for the start.
Okay. Hi, everybody. Thanks, Jason, for the introduction. As Jason mentioned, the title of my talk is Employing SSM Electrophysiology to Capture Electrogenic Fluxes in Membrane Proteins. As you can see, New York was in the headlines for a couple of weeks in the spring. It's actually a nice place. As you can see here, this is the view of my office window, looking down the Hudson River over to New Jersey. On the left side, there is the emergency room. So in the spring, there was quite some commotion going on with COVID cases. And I hope we manage to stay safe the next couple of weeks. I am actually, as Jason mentioned, I'm at Columbia University. I'm an associate professor in the Department of Psychiatry. I'm also a research scientist in New York State Psychiatric Institute. So this is the building we are looking at right now. And I'd like to introduce Eva Gill. She is my postdoc. She has performed most of the experiments. And if you have questions about experimental details, she'll be available to answer those questions. If you need to reach out to me, other than the Q&A section after the talk, please feel free to call me or email me. The address will be shown at the end of the talk again. So I want to give you some background about what I've been doing and why I was interested in the Surfer platform. So I was educated in Germany. I got my PhD at the University of Osnabrück, where I worked with transport proteins, just to start off. And I was introduced to the different principles of transport. So there's one is passive transport, and there's diffusion, and also facilitated diffusion or channel-mediated diffusion. As you can see, the model here shows a channel which is open and lets substrates, ions, whatever, through. And since I'm from a very rural area in Germany, I have a very easy way to remember what's going on here. If you have an open gate, let's say this is a field where you have a fence. If the fence is broken, the gate is open, the cows just go rushing through. There's no limit as to how many cows go through. As long as there's supply of cows on one side, they go to the other side until equilibrium is reached. As Ron Kabek said, this is a boring hole in the membrane, and I was more interested in active transport. And I focused actually on the secondary active transport processes. There was a short excursion in my career to group translocation and some ATP-driven systems, but mostly what I've been doing in the past 20 years almost, I was working with secondary active transporters. As you can see in this scheme, basically there's actually work being performed. The transport protein sitting in the membrane is opening to one side. Let's say this is the outside. A substrate and a co-transported cation, in most cases I've been working with, is coming from the outside, binding to the protein. At this time, both gates are closed, and when the internal gate opens, you have the release of the substrate and the coupling cation. This principle formulates basically the alternating access model. And again, for me, it has to be a very simple scheme. You can see here, the cow is the substrate. Cow goes into a binding site. The cowboy here is basically opening the gate. He's serving as, in my case, was mostly sodium coupled systems as the sodium ion. And you can see the wiggling of the substrate is facilitated by the action of the co-transported ion to make transport work. So when I started my work at the University of Osnabrück, I started with a system called PAD-P. It's a sodium protein transporter of E. coli, and it is sitting in the bacterial membrane. It transports proline in co-transport with sodium. When you look at uptake data, you can see that in the presence of sodium, shown here with the solid symbols, in the presence of sodium, you have uptake of radiolabeled proline. If you have no sodium around, basically uptake is similar to what you see in control cells that lack the PAT-P protein. Why do I show the sodium proton antiporter here? Because for a long time, PAT-P was considered a proton-driven system. So the idea was it was a proton proline symporter. However, the reason for this confusion was that the sodium affinity for PAT-P is so low, it's in the 30 to 50 micromolar range, that any contamination in the assay buffer is sufficient to drive transport. And this was a challenging topic to overcome during my undergrad and grad work. And the reason why the PA actually was considered a driving force is because of this sodium proton antiporter, which at low pH pumps in proton, pumps out sodium, and the sodium is available to feed into the cycle so PAT-P can work and mediate sodium-coupled proline transport. So as for a co-transporter, if this is really coupled, meaning that you have proline and sodium co-transport, you have to have the two reactions in parallel. And here what I show is up here, this is proline uptake. Here it's sodium uptake. In the presence of proline, you see sodium uptake. In the absence of proline, there's no uptake. And again, in the control cells, there's no uptake of sodium in the presence or absence of proline. For a postdoc, I went to UCLA and to Ernie Wright's lab, and I stayed in the same family. It's the sodium symporter family, and I worked with Sglt1, which is the sodium glucose transporter, which is basically sitting in the intestine and is responsible for the uptake of glucose, salt, and water into the enterocyte, which is the key system in the nutrition for people. And from my previous experiment with bacterial cells or vesicles that I made out of bacterial cells, I switched into the electrophysiology field. So we used oocytes and impaired in this the two electrode voltage clamp method. Same principle here. We took an oocyte and measured radiolabeled sugar uptake. In this case, it's alpha-methyl D-glucose, which is a homolog of glucose, just it's not degraded in the cell. And you can see that you get a substantial excess of sodium-driven glucose up or alpha-methyl D-glucose uptake in oocytes, whereas in non-injected oocytes, there's virtually no signal. And in the absence of sodium, we use choline here as the inert ion, there's very little uptake. As I mentioned, this goes into electrophysiology, and what I could see when we measure electrophysiology in choline chloride, there's very little current. So what we do here, we ramp the voltage going from -50 to +50, and then in steps to -150, and you see those very tiny currents. If you then go into the sodium buffer, which contains 100 millimolar sodium chloride, you see currents that are larger in magnitude compared to the choline chloride buffer. And there is, compared to the choline chloride currents, which basically have a very short peak and then come down to a plateau, you see transient currents here and here. And these are called pre-steady state currents, which is similar to what you see in channels as gating currents. So these transitions are considered pre-steady state currents and reflect the movement of charge in the membrane electric field. If you then add glucose or alpha-methyl D-glucose, you have a much larger current profile here with inward currents, and then at -150 millivolt testing potential, you get about like 2000 nanoamps inward current. Based on this model of electrophysiology and uptake, in the early 1990s, Ernie Wright's lab came up with this ordered 6-state model of transport, where you have basically these conditions. When you are in choline, the transporter has a transition between the open to the open outward and open inward state. This is going back and forth, but there's no charge transfer. If you add sodium, you have the phenomenon called sodium leak currents, where you have the binding of sodium to the transporter. You have a transition that opens the transporter to the inside, and then the release of sodium. And so you have this partial cycle, which is basically the sodium leak current, which is uncoupled to glucose. In this case, there's no glucose present, so this cycle can happen. And if you add glucose, once you have a sodium-bound transporter, you add glucose, the substrate for the transporter, the entire cycle occurs, and you have those inward currents that basically reflect transport of glucose and sodium. A very important number here is the turnover number. Based on these measurements, it was determined that the turnover number of Sglt is about 50 per second, which means this cycle here, from here back to here, happens about 50 times per second. In channels, this number is in the millions, so you can see the problem that's associated with working with transporters, because the turnover number is much lower in channels. So in order to get current measurements, you have to come up with some tricks and find a way to measure those currents. When I went to Columbia then, shortly after I joined Columbia University, the structure of LUT came out in Eric Go's lab. At the time he was here at Columbia too. And then shortly after, we came up with a kinetic model of LUT, which is very similar to the one I presented with for Sglt. You have basically the empty transporter binds sodium. The sodium-bound transporter can bind substrate. We have different states. We have a second binding site involved and whatnot. But the point is, you have the release of sodium is different to what we have seen in the Sglt model. But the problem with LUT is it's a very robust protein. It's almost indestructible. It's stable for months actually, and you can do amazing things with its binding and transport measurements. However, transport is very low. So leucine is a perfect bound substrate, but in terms of transport, we switch to alanine, which is a much better transported substrate. However, the turnover number is in the 0.01 per second range. So transport of LUT is very bad, if you wish to call it this way, but that's the problem with LUT. However, why do I show LUT? Because it turns out that many of the transporters I've been working with, for example, SGT1, PADP, PANF, SMBT, the multivitamin transporter, BEDP, and some other proteins, they all share the LUT fold. And here we have LUT, we have MHST, which is a protein that's better behaved in terms of transport. So we can measure actually transport, and we published a recent Nature paper where we show real-time measurements of transport in MHST. So this is my background. I wanted to give you an idea of what's going on, and switching to the surfer. So in February 2019, I went and met George at the Nanion branch in New Jersey, and I was introduced to the surfer. I got interested in this, and it was a nice way to measure currents where things got more complicated in the lab and involved radioactivity and other approaches that were not so easily feasible. So this is my first chip that I produced with George's help, and he gave me Sglt expressed in a mammalian cell line, and upon the addition of 100 millimolar alpha-methyl D-glucose, the substrate I introduced before, we see currents. So this is the initial on current, and this is the off current here. And I was very excited to see this, and it turns out that I was actually involved in the expression of this protein that was used for this experiment back in my days when I was in Ernie Wright's lab. So this was nice to see the connection. And then at this time, I just started a collaboration with Etana Padan on NHAA. So NHAA, the protein I mentioned before, which caused trouble with the measurements for PADP, is a sodium proton antiporter, and you could see that upon the addition of 20 millimolar sodium chloride, you see a nice downward current and a plateau, and an upward current comes to the baseline. In controls, which is throughout the talk shown in cyan, there's almost no current visible except noise. And I was very excited to see this because the magnitude of those currents was gigantic, and I was excited. So what I was using here was a protoliposome system where NHAA was incorporated. And the reason I was so excited because it reminded me of my days in Ernie White's lab, working with electrophysiology. So what I see here reminded me of this kind of current where you see a pre-steady state current and a steady state current. After I discussed this with Maria of the Nanion team, I understood that this is not pre-steady state current, but this is a steady state current here, and this is not a transition of the transporter in the electric field, but this is actually transported current. I was also looking at papers by Klaus Fendler, who introduced the system, and I'm kind of convinced now that these are real transported currents rather than pre-steady state currents. So this was all very promising, and as I mentioned, I'm at Columbia University. I'm also part of the COMPO team, which is the Center on Membrane Protein Production and Analysis. It's a NIH P41 supported project, and it's sitting at the New York Structural Biology Center. We produce, or we provide service to the community. So if you have any question about how to express a protein, or if you want a certain protein expressed, or if you want to have functional analyses performed, you could contact them, or if you want, you just contact me. I can connect you. And basically, through COMPO, we were deciding to purchase the Surfer N1 unit. And what do you do when you purchase a new system? You use a test bed that it's working. So in this case, we used NHAA. What's happening here is basically, as I mentioned before, NHAA is a proton sodium antiporter. It transports one sodium for two protons. In this case, we added 20 millimolar sodium chloride to the external solution, and two protons were extruded from the liposomes that were used. And you can see this downward movement, which reflects the outward movement of protons here. So the charge, the net charge that's going through here is 1, because we have one sodium going in, two protons going out, and we see only one. Why did we choose NHAA? Because, as I mentioned before, Sglt has a turnover number of about 50 per second. Some other transporters have like in the 1 per second. NHAA is a very fast transporter. It has a turnover number of about 5,000 per second. So this made this very useful and also robust system to test out the system in our lab. What I show here is basically we tested a mutant, which is aspartate 163N, and this is sitting right here at the proposed sodium binding site. This is based on the structure that was produced. However, the structure was without sodium, and based on predictions, this is the sodium binding site, and B163 is sitting right here. By mutating this one, we lose all the currents that you've seen in the wild type. We also have tested the pH dependence, but this was done by Klaus Fendler and Etana Palan in the past, so I'm not showing this at this occasion here. However, what we have done, we tested the system and we tried to get a Km for sodium. So we tested different concentrations of sodium. We started with 0.5 millimolar, 1 millimolar, 5, 10, and 20. You can see the magnitude of the currents is increasing with increasing concentration of the sodium. And as I said, what you see with increasing sodium concentrations, you get efflux of two protons, which causes the increase of the magnitude of the currents. What we have done then, we have plotted the area under the curve. So basically we filled out the section here all the way down, and also this here, and plotted the charge. So this is basically nanoamps per second, and we came to nanocoulombs. And then we plotted this as a function of sodium and reached a Km value of about 1.7 millimolar. This is pretty much to what was seen with radiolabeled sodium-22 uptake measurements in Etana's lab. The Km for sodium uptake was about 0.5 to 1 millimolar. Previous measurements I've seen also reflect a Km of about 10 to 50 millimolar. And I believe it depends on the system you're using. I have just done or completed a study with etanacodan in which we show that the experimental condition and the composition of the lipid of your liposome system is essential to maintain optimal conditions. Otherwise, you would have just partial activity in your system. The other thing I've also seen, whereas we have used the area under the curve, people tend to use the peak of the current to plot their activity. So we have done the area, but you could have also done the peak current here. So this was our test bed. Now we started the real experimental setup, and I call the surfer and malaria. What does have malaria to do with this? Actually, not much. So malaria, you know, it's transmitted by the Anopheles fly. It's not the fly, actually, it's the Plasmodium parasite that's being transmitted from the fly into the bloodstream and then starts infecting red blood cells. And as you can see here, this is a red blood cell. Inside, in this blue circle, is the Plasmodium parasite. And in this Plasmodium parasite, there's a digestive vacuole, which has a very low pH, pH 5.3. And what's happening here is that Plasmodium takes up hemoglobin from the red blood cell and transfers this into the digestive vacuole, where it's split into peptides, amino acids, and also into an inert iron complex called hemosoin. And this prevents the toxic effect of iron in the parasite. And as a consequence of this, why is malaria so deadly? Because anemia is the result of the destruction of the red blood cells, and the lysis of blood cells and the release of the parasite triggers release of cytokines, and then you have fever and sepsis. As bad as it is, in the early 1600s, it was discovered in South America that Peruvian bark can prevent cases of malaria. And then it was brought to Europe, where people were using this in more southern countries. And then before 1820s, the ground bark was used in liquids, usually in wine. And in 1820, Pierre Pelletier and Joseph Caventou isolated the compound. They isolated two, but the important one is quinine. And this was the compound that was identified. In the 1920s, 30s, new compounds based on the quinine were produced, and we have chloroquine here, and also piperacin, which is very similar to quinine. And then piperacin is basically the doublet of the chloroquine. How do these drugs act? So if you have the red blood cell infected with the parasite takes up the hemoglobin, and if you add those drugs, they pass the membrane as uncharged compounds. In the acidic vacuole, they become protonated. And in so far, whatever the mechanism is, they produce some disruption in the system to produce the hemosoin. And this process leads to an accumulation of toxic iron products, which causes the lysis of the parasite by, first of all, producing causing the lysis of the digestive vacuole, as you can see here. So these structures here are hemosoin structures. If you add chloroquine, 30 nanomolar in this case, you can see the hemosoin structures are breaking up, and also the digestive vacuole membrane is broken here, which causes the lysis of the parasite. So this is a way to fight malaria by using those compounds. However, nothing is simple. And then, of course, there's a resistance. And in 2000, David Fiddock, one of my collaborators here at Columbia, identified PFCAT protein, which is Plasmodium falciparum chloroquine resistance transporter. And why was this important? So chloroquine was used since the 1940s to fight malaria, but shortly after, like 20 years in the 1960s, it was identified that there's a resistance occurring. So you took chloroquine and you still got malaria. And David found out basically there are different versions of this transporter and he compared a wild type with a protein called 7G8. There are 5 mutations. One characteristic mutation is this K76T, which is found in all resistant forms. And you can see the drug phenotype. This one, the wild type, is sensitive to chloroquine and sensitive to piperacin. However, in the 7G8 version, which is sensitive to piperacin, it is still resistant to chloroquine. The question is, what happens? How could this be achieved? So, at the end of 2018, just after I got interested in the surfer, I got a call from Filippo Mancia here at Columbia telling me that they have a structure of PFCRT, namely of the 7G8 version. And here's a structure of this protein. If you look into the cavity, looking from down here, we see a highly negatively charged cavity, and we believe this is the binding site. And amazingly enough, we saw that a Fab, an arginine in the Fab, really sticks its finger, if you wish, right into the binding site, which was an indication for us that a positively charged substrate would bind here. So if you remember in the previous slide, I had shown that piperacin, chloroquine in the digestive vacuole where the pH is very low are positively charged. So we considered this a binding pocket for a positively charged compound. So this eventually led to the publication in Nature, and what I can show here, as I mentioned, chloroquine is negatively charged, positively charged, and piperacin is also positively charged in the vacuole. And arginine, which is an amino acid that could be a potential substrate for PFCRT, is also positively charged. So we tested uptake of these compounds. And just to make the point, this here is not PFCRT wild type. This here is PFCRT 7G8. And as you remember, this is CQ resistant, which means it transports it out, but it's piperacin sensitive, which means it doesn't transport piperacin. You can see here in this uptake experiment, time course of uptake, that no piperacin uptake is observed, whereas CQ is transported as well as arginine. This is all very nice and interesting, but for me, as a person who's interested in the molecular mechanism of those transporters, I was more interested on the mechanistic details. Why is this important? So we know these compounds are already positively charged, and the vacuole, the digestive vacuole has a pH of 5.3. So I tried to mimic the condition of the vacuole in uptake experiment. So, and it shows when we have a pH inside the liposome where we reconstitute PFCT7G8 into, the pH of the liposome is 7.5. If you go to an outside pH of 7.5, you see virtually no uptake. If you use valinomycin, which in this case produces a membrane potential, there is some uptake, but what's really causing uptake is a pH gradient, pH 5.5 on the outside, 7.5 inside, with valinomycin, which produces a membrane potential and a pH gradient. This causes uptake. So the hypothesis in this paper we just published about a year ago was that drug transport is pH coupled, and you have also a proton drug or substrate symport. The question that remained, how can we measure actually the proton flux? And there comes the surfer in very handy. So what Ava has done, she tested the effect of 0.3 micromolar chloroquine in a system where we have PFCT7G8 in the liposomes. She also played around with valinomycin to produce a membrane potential. And you can see that in the PFCRT containing liposomes, she gets a current, which is an inward current because the influx of protons is reflected as an upward current here. There is some current also in the control liposomes. And my guess is that due to the fact that CQ is not charged when it can enter, but with the low pH, it may be charged, so it may be able to penetrate the membrane. in different conditions, but also play around with the charge distribution here. When Eva used PPQ, as you can see, there was no uptake in the 7G8 version. There's no uptake. And what you see in the control, you see also in the PFCRT containing liposomes. So this was a nice proof of concept that we actually have to deal with a system that is proton coupled, performing proton coupled drug import. Next system was a uncharacterized purine transporter, which had a strange behavior. So this is binding of xanthine. And you can see it's highly pH dependent with the highest binding activity at pH 8. However, it was proposed to be a proton coupled system, and I performed kind of the same experiment as for PFCRT, having a pH gradient outside low pH, inside high pH. This is driving uptake of xanthine here. And then if you measure the pH dependence of uptake, you can see the exact inverse picture of what you can see in binding, which means substrate binds first to the transporter, and then upon the binding of protons, we initiate transport. Sorry. So the question is it pH dependent? Is it actually co-transported? And then Eva did this other experiment where she used these the liposomes reconstituted with pure T, and when she added 10 micromolar of xanthine, she saw an upward move of the current, which reflects an inward move of protons. So this is very consistent and was the missing piece of information that's now needed for submission of the paper. So most of the data I'm showing today is unpublished work, and I hope to publish this all very soon. So there was another system, and as I mentioned before, what happens many times in my life, people give me a call and say, we have a structure, but we have no idea what this protein does. Can you identify what the substrate spectrum is and how these things are working? So I call this de-orphanization. And here comes a structure from a colleague, which looks like a lutefolid protein. It is in the SSS family, so it could be a sugar transporter or a protein transporter. And based on the sequence alignment and the blast searches, it could be basically a variation of 5 proteins. So what we have done, and I'm not showing any specifics because it's very new. So Eva tested substrate A. Substrate A is one of the substrates that's the second likely candidate protein, and she got nice currency at 50 micromolar. And then when she used substrate B, which is one of the more unlikely proteins, she sees only a small signal here, which indicates that this protein, despite being assigned to a specific group of proteins based on sequence data, the functional data actually reveal the true nature of this protein. And to summarize what we have done in the past, so we used the surfer basically to detect charge transfer in reconstituted systems or cells or vesicles. We have gotten some mechanistic information about certain proteins that we have tested. We could perform substrate screening, also used for de-orphanization. We could do kinetic characterization. And the problem is when traceable substrates are not readily available or isotopes are also not available. So the problem is if you want to measure currents for the systems I'm working with, which are mostly sodium coupled, we have to work with sodium-22. And this is a major problem in terms of the reactivity that's involved and the cleaning of all the compounds and the consumables we're using. And the other problem is if you use compounds that are available or available in radio-labeled form at a very high price, if you have a custom synthesis required to have the substrate available, the surfer is actually the way to test unlabeled compounds and get initial screening data that then be used for further characterization. Again, if you want to reach out to me, email me at any time. Also, if you have any questions about Compo, you can check the website shown here. And I have not done this alone. So Ava is my postdoc and is working with the server all the time. I'm working with David Fidok in Filippo Mancha's lab at Compo. Wayne Hendrickson is the director of Compo. And with the other people, I'm working with Lei Shi at NIDA, Itana Padan, Ming Zhu at Baylor College of Medicine. And I'd like to thank George and Maria, who helped me tremendously with the setup of the server. Of course, NIH provided funding. So there's the 3 different institutes that provide funding for my work. And I thank you for your attention.
Great. Thank you so much, Matthias, for the exhilarating presentation. I'll now open the floor to some questions. So if you have any remaining questions, just feel free to drop that in the chat window. Matthias, I'll go through some of the questions now. What do the transient currents in Sglt oocyte measurements mean?
Okay, so those transients were discovered by Don Liu in Ernie Wright's lab in the late 80s, early 90s. And they came up with a comparison to charge transfer that you observe in gating currents in channels. So basically you have a charged residue or an ion that's in the membrane field that's moving based on the voltage you apply. So if you ramp up your voltage from -50 to +50, you have basically a charge transfer in the membrane that is associated with the voltage change of your system. What's happening here, it's considered a preparation of the transporter to accommodate the binding of sodium in this case. Basically, that negative charges are moved to the outside of the cell so that the positively charged sodium can bind. And then by changing the membrane potential in the 2-electrode voltage protocol, you have the movement of charges in the membrane field. These are not currents where ions move. So this is not the movable ion current where you have sodium from the outside moving to the inside. This is basically ions in the electric field moving back and forth.
Perfect. And then another question that we had, this is regarding the traces. Did area under the curve and peak give different results? And then as a follow-up, which parameters are you preferring and why?
I would prefer the area. So first of all, the first part of the question, what do I prefer? I prefer the area. We have compared area and peak. It's very similar. I prefer area because it gives us an idea of how many charges are transferred. What we're trying to do is to get an idea of how many proteins we have in our liposome system in the measurements, and to get an idea of what the turnover number is. So for this, we need the charge to transfer charge to mole using the Faraday constant. And then we can actually get an idea of how many charges are transferred per protein molecule in the system.
Okay. And then another question, can you tell me more about the membrane model? Do you need purified proteins? If not, how do you eliminate other transporters? What kind of liposomes?
Okay, so our standard approach is we have a pipeline for many transport proteins. They're his-tech, and we purify them on a routine basis. So mostly we have pure protein that's incorporated into liposomes. The liposomes are pre-made. We have basic E. coli pre-made liposomes with different additions of different lipids included. We can have some cholesterol included depending on if you have a bacterial or mammalian system. So we have a set of, like if you wish, we have a scaffold of liposomes pre-made. And then based on the origin of the protein and some experiments and expertise we have from previous experiments, we then choose a certain set of liposomes to incorporate the membrane protein. We have tested a little bit in vesicles, and I think part of the question is, yes, there is background because there is always mostly an interfering system that you have some noise in the system. With reconstituted membrane proteins in the liposome system, this noise is greatly reduced.
And then another question that came in, you mentioned chloroquine and hydroxychloroquine. This has been in the news a lot lately. Do you think this can work in COVID-19 in a similar way to malaria?
As far as I understand, chloroquine and hydrochloroquine affect a acidic vacuole where the virus is captured at the very beginning of the infection. They may be able to collapse the acidic vesicle at the time of infection, but I don't think it's a long-term treatment, and I don't think it's very promising. I was afraid of this question, and I'm not an expert in the virology field, but I would be very careful of making the transition from malaria to COVID.
And then as a follow-up, do you see other transporters that might be targets for other diseases?
Yes. I mean, there has been, so for example, COVID is known to target a certain protein, and we just realized that this protein is interacting with a transporter in the LUT structural fault. So there is a connection between different proteins, and if they are needed to reach the membrane, if they're needed to function properly, there is a connection. And I believe, yes, transporters are a very promising drug target.
And then another question that came in your de-orphanization project, the current of substrate 8 looked very steep. How do you interpret this?
This one here. That's a good question, actually. We have seen other currents. So this is one trace of the currents. We have seen other currents that are a little bit more broad. So I didn't pay too close attention to this, I have to say. But
we can also, if you need more time, we will transcribe this. So I know that it's a little difficult to do it on the spot. I'm happy to include this in the Q&A. I mean, we have other questions that came in as well.
Okay, I honestly don't know the answer to this question right now, and I'm not sure if I can answer this later. I'll take a look into this, and I'm happy to answer.
Okay, okay. Yeah, any commentary that you have on this, I'm sure would be much appreciated. You mentioned the liposome composition has a huge influence on transporter activity. Which liposomes do you prefer?
The ones that work. So, if I could have shown the previous presentation, my problem was always, so you have a membrane protein, you produce it in the system, and then you extract it, and then you work with purified protein. Then you go into crystallization, or you go into function studies by incorporating this into liposomes. For me, the step between the native system, expression system, to your assay system, be it pure protein or in an artificial membrane system, was the big question mark. What's happening there? So I have used the SPA, the scintillation proximity assay, to monitor the activity of membrane proteins in detergent solution. I could see that different detergents caused effects on proteins in terms of binding. I mean, the craziest thing was the whole LUT story, where we identified that the detergent octoglucoside used for crystallization of LUT was actually binding in a site that's important for substrate transport. So what you see in the crystal structure is a impaired protein because it has a molecule of detergent sitting in the critical part of the protein. So by moving away from crystallization, using detergents and using all kinds of different detergents and amphipoles and whatnot, and going into more cryo-EM structural determination, you move away from this kind of artifact. But also, if you do cryo-EM, you have to make sure that your membrane environment has to be close to the native environment as possible. So we have, as I mentioned, a scaffold of different pre-made liposomes. And if something comes out of bacteria, we usually go with the E. coli total or polar lipids. Then we add certain additives, be it cholesterol, be it POPC, POPG, cardiolipin. All these things were added to those basic formulations, and then see how this works. So it's like you test a certain system. If this works in your liposome system, you add something based on experience. There's no final answer. There is no perfect liposome for all systems. There is a perfect liposome for system A and a perfect liposome for system B. It has to be tested out.
And then another kind of question to tag along more on the technology side. How does the surfer compare with data from the radioactive methods you've used? Or how does the experiences with the surfer N1 compare to other methods you use to record transporters?
Very nicely, actually. So I have seen, I was, and I have to say, I'm still a little bit convinced that the best way is the direct way to measure transport. So if the system is sodium dependent, I would like to see sodium-22 measurements and compare to the surfer. But I'd like to do this once and then compare with the surfer measurements and then have the system established for all subsequent measurements. I have seen in many cases where fluorescent dyes were used to measure either sodium or other ions entering a cell or a liposome. And the readout is an order of magnitude, or the constant you get from this kind of readout is an order of magnitude higher than what you see with other measurements. For the surfer, I have done recently NHAA uptake with sodium-22, and the affinity is in the 0.5 millimolar range. For the surfer, it was 1.6. This is like a 3-fold difference is, in my opinion, acceptable. I have seen previous measurements on surfer where the affinity for sodium was in the, I believe, 10 to 20 millimolar range. So it also depends on the orientation of the liposomes. And then for NHAA, I also saw there's a recent paper by Etana Padan's lab where you can see that the composition of the liposomes and the environment of your protein in detergent solution affects the activity of the protein. So in this case, high concentration of detergent is bad, as well as the lack of cardiolipin. So if you reduce the detergent concentration and add cardiolipin into the system, the activity of NHA is much better. I don't know exactly how the experiments were done in the past, if the protein was exposed to non-optimal conditions in terms of detergent and lipid, what the liposome composition was. All these factors play in. So I'm very careful about the environmental system I'm working in, what the readout is.
And then a follow-up to this, regarding protein properties, do you think you can investigate with the surfer N1?
Could you repeat this, please?
Sorry, which protein properties do you think you can investigate with the surfer?
Oh, I believe you can investigate if a protein transports ions. We also have tested some channels. We get huge currents with this one. We found some differences from what was seen before. I believe because nobody tested, so we played around with this. So I believe you can see the basic feature of charge transfer through a protein. I believe once we established a stable baseline, we can then test stability of the protein, effect of detergent, the effect of liposome composition. I believe this is all a way we can use the surfer for.
And then following, again, more about this, what kind of results do you expect from electroneutral transporters?
There's the problem. So if there's an electroneutral system, there has to be a way to block one of the countercharge movement if possible. But if this is part of the main mechanism, I think there's a problem. I mean, there has to be a net charge movement one or the other way. Otherwise, we won't be able to see this. For example, the sodium iodide transporter, you could nicely see sodium and iodide movement because the stoichiometry was 2 sodium per 1 iodide. So you had a net charge of 1 positive sodium going into the oocytes in this case. But if perchlorate was used, the stoichiometry was 1-to-1 and it looked as nothing would happen. So perchlorate was not considered a transported substrate, but was a blocking compound. But it turned out after like very elaborate experiments, very nicely done by Nancy Carrasco's lab, that perchlorate is transported. So they came up with a very elegant way to detect this. I don't think that with the surfer, this kind of assay would have been possible. And I think this is the gap where you really have to go back to radioactivity and fluorescent measurements, maybe.
And then lastly, how can you say by analyzing the transients that the protein transporters are not only a binding event?
With the transients, I cannot rule out that this is only a binding. So transients can reflect binding. That's true. I think what's important, if you look at the currents, let me just go quickly up there. Sorry. There we go. So the transient currents may reflect binding. However, what's important here that is actually transport are those. If you, I have to go back another slide. So if you compare to the blue line, there's very little current here, which is just leak currents through the experimental system, the oocyte here. With the white currents here, you can see that the baseline here at minus 50 millivolt is moved down a little bit. Basically this line here. And these transients could be binding, unbinding of sodium. This could be exactly this, well, I guess this step. This is basically going from the empty transporter to the sodium bound transporter. And this arrow is bidirectional, so I can go back and forth, basically on the membrane potential. So if you have a potential that allows the empty transporter, you would see very little transient currents. But if you have binding of sodium and unbinding based on the pulsing here, you can see those transient. These currents here, these steady state currents, these are actually transport from going from this conformation to the other conformation. And again, if you change the voltage, you can also have the reverse step. So this cycle, this partial cycle can go back and forth depending on the membrane potential here, where you have binding, unbinding, and actually transport.
Perfect. Once again, thank you so much, Matthias, for joining us today and giving us a nice look into your work. I'll make sure that the transcribed copy and on-demand copy of this recording is made available more likely than not tomorrow morning at the latest. And I'll make sure to tag along all your contact details for you and your group. And naturally, anyone interested in the technology can reach out to us and yourself for references as well. So just want to wish everyone a good rest of their days, and we look forward to tomorrow. There's a heavy schedule, so join when you can.